For developing nucleic acid therapeutics, it's absolutely critical to understand the innate immune receptors that detect foreign nucleic acids so that you can avoid unintentionally triggering inflammatory and other immune responses to your therapeutic. On the other hand, activating these receptors in a controlled manner can be very useful. These nucleic acid immune sensing receptors evolved to detect infection, especially from viruses. And agonists to these receptors can activate innate immunity, which includes inflammatory cytokines and type 1 interferons, and that leads to the generation of adaptive immunity, especially antibodies and T cells. And these adaptive immune responses can be therapeutically very useful. Agonists to these receptors can enhance vaccines, as we'll discuss in this video, and cancer immunotherapy, as we'll discuss in an upcoming video. This table shows the nucleic acid immune sensing receptors that are being targeted or have been targeted in human clinical trials. Some of these receptors recognize DNA and some of them recognize RNA. The DNA recognition is accomplished by TLR9, toll-like receptor 9, which is in the endosomes of cells, and cGAS sting, which is a cytoplasmic receptor pathway. Now for TLR9, the agonist is unmethylated CpG DNA. CpG is cytosine phosphate guanine. And in our genomes, CpGs are methylated and they're suppressed. And so unmethylated CpG, which is present in viruses and bacteria, is a specific ligand that's a sign of infection. Now the cGAS sting pathway in the cytoplasm can be activated either by double-stranded DNA or by synthetic cyclic dinucleotides. But it doesn't distinguish foreign DNA from self-DNA. This pathway is not normally activated in cells because DNA is normally sequestered in the nucleus. If cells are stressed or if apoptotic cells are taken up, then the pathway can be activated. With RNA molecules, the same concept applies. Several of the receptors are present in endosomes, and two of the receptors, TLR7 and TLR8, recognize RNA sequences containing uridine, either polyuridine or GU-rich RNA sequences, that make it possible to distinguish foreign RNA from self-RNA because the most abundant cellular RNAs, ribosomal transfer RNAs and small nuclear RNAs, use pseudouridine instead of uridine, and pseudouridine doesn't activate this pathway. Likewise, in the cytoplasm of cells, there's a family of receptors called RIG-I-like receptors, and RIG-I is activated by RNAs that contain a 5' triphosphate or diphosphate, whereas in most of our cellular RNAs, the 5' end is monophosphate, and so does not activate RIG-I. Another way to look at this for the RNA sensing immune pathways is that foreign material is taken up into a cell, bacteria, viruses, apoptotic debris, or synthetic RNA, and when that material enters cells, it's normally taken up into an endosomal compartment where if it has the right structures, it can activate one or more of these receptors. Now there's two major downstream pathways that can be triggered. One are the interferon response factors leading to generation of type 1 interferons, and the other major pathway that can be stimulated is NF-kappa B, leading to inflammatory cytokines. Likewise, in the cytoplasm, we have RIG-I and other cytoplasmic receptors that again can activate interferon response pathways or inflammasome pathways leading to other inflammatory cytokines. And the same concept applies for DNA. Material taken up into cells that contains DNA or synthetic DNA nucleic acid therapeutics can activate TLR9 in the endosomes if it contains unmethylated CpG DNA especially. And in the cytoplasm, there's the cGAS sting pathway that can be activated by endogenous DNA if cells are stressed. So this pathway also can provide a way to maintain cellular homeostasis. And again, the downstream signaling pathways can lead to generation of type 1 interferons or to cytokines or to both. as well as to the inflammasome pathway in the case of sting. Now let's focus on TLR9. In humans, there's really only two immune cell types that constitutively express TLR9. One are B cells, and the other are a type of dendritic cell called plasmacytoid dendritic cells. And so when we add CpG DNA in this situation, these cells are activated. The plasmacytoid dendritic cells are the main cells that produce the type 1 interferons, especially interferon alpha, and we'll come back to that. They also can secrete exosomes containing antigens that they have taken up around them. And then downstream from that, the plasmacytoid dendritic cells secondarily activate other cell types, including natural killer cells and other classes of dendritic cells. B cells secrete inflammatory cytokines and also express co-stimulatory receptors that makes it easier to activate them to secrete antibodies if they also receive a signal through the B cell antigen receptor, if they bind the antigen that they're specific for. And then downstream from that early innate immune response that occurs in the first few hours is the adaptive immune response that occurs over the following days and weeks. And that adaptive immune response, in the case of B cells, leads to the secretion of antibodies that are specific to the antigen that the B cell was stimulated with. The plasmacytoid dendritic cells, together with other dendritic cells, induce antigen-specific T cells, including CD4 T cells that help the antibody production, and in some cases, CD8 T cells that can attack infected cells, or this is the cell type that can be harnessed for cancer immunotherapy. So let's talk about using a CpG DNA to enhance a vaccine, where you really want to maximize the antibody response to the vaccine. How well does this actually work to improve a real vaccine? And the first case where this was tried was with a hepatitis B vaccine called Engerix B, which is an approved hepatitis B vaccine. Engerix B uses an adjuvant called alum, or it's a type of aluminum salt that was developed 100 years ago to enhance immune responses. And adding alum to the hepatitis B surface antigen leads to the production of antibodies that can protect against hepatitis B infection. The approved regimen of vaccination with Engerix B is 3 doses at the baseline, the first dose, then a month later a second dose, and then 6 months later a third dose. And after that third dose at 6 months, about 80% of people normally will achieve protective antibody levels that are known to protect against infection with hepatitis B. So what happens if you add a CpG DNA in place of the alum adjuvant that normally is used with that vaccine? Well, this is called Heplisav-B, which contains 3 milligrams of a synthetic CpG oligonucleotide with this sequence. It just has 2 CpGs in it. And what you can see that is a very dramatic finding with the Heplisav-B, there's a huge acceleration in the development of the antibody response. It only takes 2 doses for more than 80% of people to achieve protective antibody levels. So at 2 months after the first dose, instead of after 6 months, most people are protected, and that's a higher percentage of people being protected. So this was approved by the FDA in 2017, and since then, post-marketing studies in tens of thousands of subjects have confirmed that there's similar safety of Heplisav-B compared to Engerix B, and so I think there's a great deal of potential for other vaccines also to benefit from the substitution of CpG DNA for other adjuvants to accelerate and enhance those antibody responses. And in the next video, or in an upcoming video, we'll talk about the application of CpG DNA for cancer immunotherapy as well.